let's discuss the fourth enzyme of importance in different pathological situations, and this enzyme is alpha 1 antitrypsin. So, what happens that in different pathological bacteria or viral precursor proteins, they get processed by the human proteases. It is sort of ironic but true that they are non-infectious or non-pathogenic to begin with when they are in their earlier precursor stages. But when a human protease acts on them, then it converts those inactive precursor into active potent protein of pathogenic qualities. And those proteases, human proteases, they recognize a specific peptide sequence, and that is arginine. And then the next one could be anyone, followed by a lysine or arginine in the third position, and in the fourth position again arginine. So arginine, next, first position arginine, second position any amino acid, third position lysine or arginine, and the fourth position arginine. Um, that is the 4 amino acid long peptide which is recognized by the protease enzyme. Fine. And after these 4 amino acids in any polypeptide chain, the protease cleaves. And when the protease cleaves out this segment, it ultimately converts the inactive precursor into the active potent precursor. So this processing step is common to several infectious agents. A therapeutic agent that targets this processing enzyme and blocks the activity of the protease, which might turn the inactive protein into active one, will be termed as the broad spectrum antibacterial and antiviral agent. So, keeping this in mind, the scientists actually worked on the expression of alpha 1 antitrypsin enzyme. So that was genetically engineered and tested in tissue culture experiment. And that enzyme was found useful in blocking the processing of human immunodeficiency virus, which is HIV, type 1 glycoprotein, which is also known as GP160. It also blocked the processing of measles virus protein F0. It also blocked the production of such proteins in many other infectious viruses. So when alpha-1 antitrypsin variant was added to the cell cultures, it blocked the production of human cytomegalovirus. Cytomegalovirus is the one which is responsible for the failure of organ transplant in the recipient and the AIDS patients. So that is a kind of very, very important and a huge scientific discovery. Let me tell you one more thing, that the alpha 1 antitrypsin variant is both potent and selective. Means not only that it will specifically block the protease enzyme, but it will highly, very, very efficiently block it. So there is at least tenfold more effective and potent and selective inhibitory action when we use the recombinant alpha 1 antitrypsin variant than any other viral inhibitory agent. Its efficacy has been demonstrated in the cell cultures, which means although it has proven very effective in the cell culture, but we still don't know whether it will give similar result in the whole animals or not. So this study remains to be done and it is under process. The next enzyme to be discussed is glycosidases. And the idea behind using this glycosidase is very interesting. You must have heard of the need for universal donor blood type in case of medical emergencies, in case of road accidents. So the blood bank requires a lot of universal donor blood type, and I hope you all are well aware of that the universal donor is O, and the AB group is considered as the universal recipient. Now, the idea behind the whole story is that the difference between the blood groups A and B is because of their side chains. If those side chain or those side carbohydrate moieties can be removed from the surface of the erythrocytes, endothelial cells or some other cells, then they will be turned into O type blood cells. So even if we have blood type A or we have blood type B or we have AB, if we can treat them with specific glycosidases which remove the side chains, then they will convert into the O type blood group, which can be given to any blood group patient. It's because it is universal donor in nature. Fine. So I hope you all really appreciate the problem and the solution too. The scientists actually screened 2,500 fungal and bacterial isolates for figuring out the right glycosidases, and they found an active alpha N acetyl galactose amidase enzyme which converts group A to group O in a gram-negative bacterium, Elizabethingia. So they isolated that enzyme from that bacteria, and it was found useful in removing the side chain in the type A blood group, thus converting into type O blood group. They also found alpha galactosidase A, which converts group B into group O, and that enzyme was identified in Bacteroides fragilis, which is a gram-positive bacteria. So their genes were isolated and their proteins were characterized, and you can see a very interesting scientific publication here, an enzymatic pathway in the human gut microbiome that converts A to universal O type blood, which was published in Nature Microbiology, and it is really worth reading. That is why I want you to go through it and make a flow diagram of its materials and methods.